All right, let's go ahead and get started. So thank you so much to the St. Baldrick's Foundation for giving me the opportunity to present to you all today. As I said, my name is David Higgins. I'm here representing the Gabriella Miller Kids First Data Resource Center. And I'm excited to tell you about the data sets and platforms that our center supports in order to be able to help support pediatric cancer research. So as I said, I'm here on behalf of the Kids First DRC, and I do want to submit my or share my acknowledgments here right at the very beginning before diving into the talk. So the Kids First DRC is a huge collaborative effort across 5 different institutions. It's the work effort of dozens of people, most of whom are represented here. So I wanted to give them all an acknowledgement and a shout out. And as you're after the talk, I please encourage you to visit us at kidsfirstdrc.org to learn more about Kids First. You can follow us on all social media platforms at Kids First DRC and reach out to us, support@kidsfirstdrc.org is our support inbox and the best place to get in contact with us after talk. So specifically, my name is David Higgins, as I am here representing the Kids First Data Resource Center. I'm employed by the Children's Hospital of Philadelphia, where I'm the Informatics Program Manager. Part of my job is working on the Kids First Data Resource Center's operation team. And so here we've got some of our team members who do a lot of the effort to generate the files and that I'm going to talk about here today. So on the top row, that's our data operations team. That's Christina Diaz, Rima Williams, and Amanda Warco. And then on the bottom, that's 2 members of our bioinformatics team. That's Bo Zhang and Brian Ennis. And of course, many more people whose efforts have gone into our work today. So today, at today's talk, I'm going to talk a little bit of an introduction about what is Gabriella Miller Kids First, what resources are available for pediatric cancer researchers, and then I'll end with some live demonstrations where we'll be building a cohort of participants on the Kids First portal. We'll review files and learn how to apply for access, and then we'll analyze data in the cloud on Kavatica. All right, so starting at the beginning, what is Gabriella Miller Kids First? And so the very kind of best way to unpack this is to start at the very beginning. And who is Gabriella Miller? Gabriella Miller is a little girl. She was diagnosed with diffuse intrinsic pontine glioma, a DIPG, which is a type of brain tumor. Unfortunately for Gabriella, she passed away less than a year after her diagnosis. But in that year, she spent a lot of time becoming a very outspoken self-advocate for pediatric cancer research. And here she is at an event that she helped organize. And one of her famous quotes that I think about very often here at the Kids First DRC is when she was requested to ask, what would you tell people in charge or people in the government or people in Congress about pediatric cancer? Gabriella Miller said, "Talk is BS. We need action." So yes, Gabriella, I 100% agree with her philosophy, and we use that to guide us. Talk is BS. We need action. We were able to act through Gabriella's efforts. And so a law was named after her, the Gabriella Miller Kids First Research Act, which was signed on April 3rd, 2014. It's actually a picture of the signing, as well as Gabriella's mom and her younger brother in attendance there. So what this act does is it transfers money into a pediatric research initiative fund, and then it supported, spread over 10 years through the NIH Common Fund for Pediatric Research. So what this act unlocked is that it unlocked the formation of the Pediatric Research Program. This is an NIH Common Fund initiative, and their goal and this NIH-funded office is to be able to facilitate research into the uncovery of new insights into the biology of childhood cancer and structural birth defects, including looking at the overlap and any shared genetic pathways between those 2 conditions. So this is a very big effort. It requires a lot of coordination and generating genomic files and clinical data in order to be able to develop it and facilitate this research. And so the program has entrusted us, the Kids First Data Resource Center, to carry out the operations necessary to complete those objectives. So this is the Kids First DRC. Again, we are a partnership of more than 5 different institutions, and our goal is to process and analyze data sets, develop web platforms to host those data sets, and then facilitate collaboration and use of them to support pediatric cancer and structural birth defects. So our goal, the Kids First Data Resource Center, we're a collaborative pediatric research effort. We want to understand, as I mentioned, the genetic causes and links between childhood cancer and structural birth defects. So I've mentioned that a couple times now, the link between cancer and birth defects. So why should we study birth defects and pediatric cancer together? So cancer is the leading cause of death by disease in persons younger than 20 years of age in the United States of America. However, children with birth defects are more likely to be diagnosed with cancer before they turn 18. And so children with non-chromosomal abnormalities, these are things like heart defects or cleft lip, are 2.5 times more likely to be diagnosed with cancer in their first 20 years of life. Children with chromosomal abnormalities, these are things like Down syndrome or trisomy 18, are 11.6 times more likely than their peers to develop cancer before they turn 20 years old. So here's some figure from a recent research paper looking at that overlap. So this is a heat map indicating the overlap of cancer type and structural birth defect with different ratios of increase there. And so what this is telling us is that there is a common underlying pathway that might be connecting these seemingly unrelated rare diseases. So even though a cleft lip and a neuroblastoma seem to be very different, it's possible that the same developmental biological pathway might be causing these two different diseases. And so therefore, by studying them together and combining them, we can empower our analysis to be able to learn more rapidly. And so what the Kids First DRC does is we work with clinicians and researchers around the country who are all domain experts in their particular field. They've collected samples, they've consented patients and participants in clinical studies at their particular institutions. What we do is we work with them who are all experts in their particular study and their particular field, and we provide high quality clinical and genomic outputs to help them analyze their project further. To do that, we partner with sequencing centers who are delivering and producing these sequencing genomic data sets that we then pair with harmonized clinical data. We would deliver these resources back to the original research group, but then we also release them out to the general public, to secondary users and researchers who can then continue to try to search through the data and make new discoveries and make new breakthroughs for these children, for these patients. So our data resource, so this is just an overview of data that we've released so far. We've released more than 55,000 files, representing more than 13,000 participants and more than 1.5 petabyte of data. Each of these studies listed here has harmonized clinical diagnoses, phenotypes, and demographic information that's all searchable on the Kids First portal, which we'll see here in a little bit. But we're not done. We have a long way to go. And so, in fact, we've been funded for additional 21,000 samples have been funded and are in the process of being sequenced, delivered, and then ultimately released on the Kids First portal as well. So these are coming soon, you could imagine. All right. So I'm here today speaking at the St. Baldrick's Foundation. And so specifically, I wanted to talk about what resources are available for pediatric cancer researchers through the Kids First DRC. And so our data resources in our pediatric cancer studies are shown here. And so each of these studies are released and available today. Nearly 11,000 participants across these different studies are all available on the Kids First portal today. So these include the Children's Brain Tumor Network study, which is a large collection of pediatric brain tumors across multiple different histologies. We also have several leukemia studies. We have a Ewing sarcoma and osteosarcoma studies, as well as neuroblastoma as well. Additionally, we have other more general cancer studies as well, our intersection of cancer and structural brain defect study, as well as our cancer susceptibility study. So we have a large collection of different genomic resources across with enrolled patients across these different studies that are available for investigators today to access. Coming soon, again, in the release queue, we have some additional studies coming soon as well. We have some germ cell tumor studies, as well as a rhabdomyosarcoma and additional bone tissue and pediatric AML data sets as well coming soon. So keep an eye out for that. So for each of these cancer studies that are available today, we offer harmonized genomic files. And so we make this available in numerous different formats. And so we try to empower researchers, depending on where they are and how supportive and how whatever types of skills and expertise they have in order to be able to access this type of genetic information. And so investigators who are interested in starting at the very beginning, maybe you have your own bioinformatics skills, you have your own pipeline that you're interested in running samples through. We offer at the very beginning, we offer the aligned genomes. And so you can start from the very beginning and run your own pipeline. But for investigators who maybe want to work on data that's already been processed a bit, we've begun those first steps. So we've called germline variants in each of these participants. And then in additional studies, we've done additional processing as well. So for example, in our trio-based study, we'll do a family-based joint call. So what this is a trio-based study, is a study where there is the child patient was consented, but then also mom and dad as well also consented to join the study and contributed a DNA sample. So what we'll do is we'll actually combine all three of those samples together into a joint-based analysis, so you can track what variants were inherited from mom, what came from dad, what new de novo variants might have arisen in the patient by themselves, or what combined together from mom and dad. And so it allows you to be able to track things within a single family, all combined together. For each of our cancer studies where we have tumor data, we also do tumor normal comparison as well. So these are these somatic variants. These are specific mutations and variants that are arising strictly in the tumors themselves. And so we have multiple different outputs from our somatic variant pipeline. So we have single nucleotide variants, copy number variants, and structural variants, all in two formats, both BCFs and MAPs. And so some of our studies also have RNA sequencing data as well that's paired with it. And again, we offer a range of different outputs for investigators to be able to use. So if you're interested in running your own analysis, we make the unaligned FASTQs available, but we also have done our own RNA sequencing analysis ourselves and generated expression counts and gene fusions, which are also accessible. So I mentioned at the top, these are harmonized genomic files. And so I use that term, so I want to define that really quickly. So harmonized, what I mean by that is all of our different studies, if I go back, all of our different studies listed here have all been run through the same workflow. And what this does is it allows for cross-disease comparisons and analyses. So you can combine participants from the multiple studies, even though they were consented elsewhere under different protocols. They can now be combined together to empower larger cross-disease comparison and analysis and discovery. So we make the outputs, we make these files, the samples, the genomes, the variants, we make all of these files together, but we also make the tools that generate those files available as well. So we have four main, what we call pipelines. We have our alignment and haplotype caller pipeline. This generates the CRAM files and the GBCFs. And then we have the three additional pipelines that I mentioned, the trio-based joint genotyping, the somatic variant tumor normal, and the RNA-seq pipeline. And so each of these is available on our platform Kabatica, which I'll demonstrate here in the second half of our talk, as well as in our GitHub repository for investigators to learn more about. And so what our intention is not to keep anything hidden or mysterious. We want to be very transparent with investigators. We want to make our tools available for anyone to be able to use, and especially if you're interested in bringing your own data. So if you have your own set of genomes that might have been funded with a different study and you want to combine with the Kids First data set, we want to make it very easy for investigators to be able to bring their own data, run your files through our workflows, and then your data is now harmonized too. And now it has interoperability with those data sets that I described previously. So where are all these files housed, and where can we locate them? So these can be found on two of our Kids First cloud-based platforms here. So the first is the Kids First Data Resource Portal. So this is kind of our search engine, if you will, allowing investigators to be able to explore the different data sets and build cohorts of participants. Once you have those set of participants, then you can look up the particular genomic files and identify those to be able to use those for further research, and then connect these data from multiple Kids First studies, again, relying on that harmonized output that we generate across all of them. Once you have all those files, we make it available for investigators to export them or download them to a local computer or a local high-performance computing system that you might have access to. But we strongly encourage folks to use Kivatica, which is our cloud-based analysis platform. And so rather than doing the computation locally, we encourage people to do the computation where the genomes and where the files are up in the cloud. So Kivatica allows users to be able to do these large-scale workflows and compute on these genomic files. And so all of our files are actually generated in Kivatica, and we use it for our bioinformatic analyses ourselves. But of course, once you have the list of variants, the project is at that point kind of just beginning, because then you need to analyze the data. You need to look and see, what genes are represented here, what is inherited. And so you can analyze the data in the cloud as well, using interactive nodes and accessing via RStudio and Python notebooks, so you can review the data within the cloud itself. And then finally, Kivatica is excellent for sharing both tasks and sharing findings with collaborators around the world. So investigators who are working on an institutional system might not always be able to provide their collaborator at another location access to that internal system. So what Kivatica does is it makes those sharing easier. Just by adding a particular user, you can invite them. They can see everything that you see right in your project. And so kind of the workflow which we'll go through here in the second half is that we'll begin with our inputs. So we'll select some files from the Kids First Data Resource Center portal, and then we will move them into Kivatica, where we'll be able to review different workflows that we can use from our existing library, or ultimately, which can then ultimately lead to our data cruncher. We'll be looking at the files in a little more detail. All right. So at this point, I'm going to move into kind of the second half of our presentation. We're going to go into a live demonstration of some of our resources. So I'm going to start by visiting the Kids First portal, and we're going to browse participants, and we're going to build our own virtual cohort, identifying participants in a couple different ways. After we do that, we're going to review the associated files with those participants, and then we're going to apply for access. And then once that is complete, then I'll show how to analyze the data in the cloud on Kivatica and the structure of what that looks like. All right, at this point, I'm going to exit out of my full screen, and I'm going to pull up a new tab here. We're going to navigate to portal.kidsfirstdrc.org. And so if you're navigating to the portal for the very first time, it will take you to this page where it asks you to either log in or sign up. At this point, if you click to sign up, it will ask you to connect using a couple different options. So anyone with a Gmail authenticated ID can log in. Any Gmail address will work, as well as if you have an Org ID, then you can use that to log in and create an account. And also if you have an NIH RAS ERA Commons ID, you can use that as well. So anyone can create an account and look through the files that I'm going to show today. I just want to emphasize that you don't need to be, have the NIH identification. Oops. Let's go back to our homepage here. So I already have an account, so I'm not going to create an account today, but if I needed to create one, I'd click sign up. So I'm going to click login, and I'm actually going to log in with my RAS ID. So now I'm in. So once you enter the Kids First portal, you land on our what's called dashboard. And so our dashboard provides a couple different widgets here that provide overview information about different parts of the Kids First portal. So let's go back to our slide here. So we're interested in browsing participants and building a cohort here. And so there's a couple different ways to do that. And so the first thing we're going to do is we're going to identify participants by study. So the best way to do that is to go to the second tab right here, which says Studies. We're going to click on that. And when it loads, it takes us to the study page. And so all the data within the Kids First portal is organized in this concept called studies. Each of these studies has a specific shorthand code that we use, but it also has a longer form name, which is a little bit easier to understand. Within these particular studies, we have links out to dbGaP. This is how we'll receive access to the data a little bit later, as well as overview information about the number of participants, number of families, and different types of data that's available for those particular studies. So if we're interested in cancer here with the St. Baldrick Foundation, then we can select Show Me All the Cancer Studies. And so you can do that by going to Domain and choosing Cancer. And so you can see now I've added a filter here up at the top. We're looking just at our cancer studies, and it's narrowed us down to 15 studies. So for example, we can then see by name, the Kids First Neuroblastoma study, the Kids First Myeloid Malignancies study, Kids First T-cell leukemia study, and so on and so forth. And so you can see if you're an investigator who's interested in a very specific diagnosis, you can use this as a quick way to be able to find data sets that might be interesting to you. So perhaps if I'm an investigator who's researching neuroblastoma, I could click KF-NBL, and that will take me here to the Explore Data tab where I can dive in a bit deeper. All right, and so on this tab, so now we've kind of moved to the third tab here. This is Explore Data. So what Explore Data is it's a little bit like a search engine that you might use at a library. And so if you're looking for a specific book at the library, you could imagine you would type into the catalog, show me just books by this author. Show me books that are published in this year, show me books with this title, and then the library will narrow that down to hopefully get you to find just the book that you're looking for. So it's similar with the Kids First portal, except we're searching across enrolled participants in Kids First studies. And so we're looking at this point, we've already applied a filter. We've applied this, participants in the NBL, the neuroblastoma study. And so we're looking at 1,600 participants and 600 families and more than 18,000 files. It gives you some additional information about different data types that are available, a number of additional diagnosis information, as well as some overviews of demographic information as well. So we came here again from the Studies tab, and then we moved over by clicking that link out. But we could also look up participants in additional ways as well. So I can start a new query, and you'll see on my bar charts I've reset. So now we have kind of removed that filter, and so we've zoomed out a level. And so this is at our widest possible part of the funnel here. We have more than 30,000 participants in here. So we want to apply some new filters to find participants that are relevant to us. And so, for example, I'll show you. So we came at this by just looking at the participants in the neuroblastoma study, but it can be more advantageous to identify participants by clinical phenotype as well. So if I go back to our portal, then I can search for participants who are labeled with the diagnosis neuroblastoma. I'm going to type that into our search box. And the search box pulls up under diagnosis Mondo. We're going to apply this filter here. So just show me participants who are tagged with neuroblastoma. And so now you can see we have 800 participants in 800 families. So these are specific participants with neuroblastoma, not just their parents. So we see across the neuroblastoma study as well as the target neuroblastoma study. So that would make sense so far. But then additionally, we see, oh, there are other patients in the intersections of cancer study, as well as the pediatric brain tumor atlas study and the novel cancer susceptibility study. And so by coming and searching from this diagnosis angle, we can apply and find more participants, more possible studies that might be relevant to our analysis, to our discovery. So that was adding a filter for diagnosis, but I can actually go through and apply one more filter. So I'm going to use one of our clinical filter tabs, the observed phenotype, HPO. So HPO is human phenotype ontology, which is a set of an ontology of terms used to describe different observations about participants. And so we also have a lot of, as our, Kids First is pediatric cancer and structural birth defects, we have many structural birth defect observations as well. And so, for example, if I type atrial septal defect, which is a heart defect, we can search for participants that have this particular phenotype as well. The nice thing about HPO is that it's hierarchical. And so you can select very, very general terms, abnormality of the cardiovascular system. You could imagine there's hundreds or thousands of different abnormalities that would fit that category. Or you can select very, very specific. So not just this particular heart defect, but even more specific than that. So we're going to stick with the larger, higher level term here, atrial septal defect, and we're going to apply that. And so what happens is that it gives us information about, again, applying this filter. So now we're looking at participants who have this heart defect, the atrial septal defect. And again, the kids in the heart defect study are two that come up as perhaps the most, but then you can see here also in our other studies, there are other children born with this defect as well. And so therefore we can begin looking at combining these different analyses together. So maybe I'm interested in the intersection of these two. And so maybe I'm really interested in looking at, well, do any of these kids with neuroblastoma and any of these kids with the atrial septal defect, are any of them the same? And so you can actually combine these studies together. So by, let's say, I want 2 and 3 combined. So the answer is yes, we do find that. In fact, there's 2 participants in the pediatric brain tumor atlas who were diagnosed with neuroblastoma as well as atrial septal defect. So it gives you this very kind of specific output. And now we're just looking at 2 participants. So we've gone from thousands all the way down to a much smaller number. You can click over to table view, and this gives us more information about these particular participants overall, as well as their diagnoses. At this point, though, if I want to include, maybe I'm really interested in these participants, I could then look at the files to learn more about them. So I'm going to pause this right here because before I move on to the files, I want to search one more way, and I want to show one more tool. So you can also identify participants by genetic variant, which I'll show right here using our variant search tool. So our variant search tool is our 4th tab up here at the top. So if we go over to variant, brings us to variant search. So what variant search is a search engine where you can imagine explore data, you're searching and you're filtering based on clinical and diagnostic information. Variant search, you're filtering and searching based on genetic information. So these are germline variants that we are searching for in our search bar here. So there's, this is across all of our different, across 19 different Kids First studies. We have about 11,000 participants in here, and more than 309 million variants and 61 billion occurrences. So lots of variants in here. So how do we search across them? So you can search by applying a filter. One of our tabs of filters in here, I like to typically start with gene, and we can search for a gene of interest. So since we're talking about neuroblastoma, I might be interested in the ALK gene. Click apply. And so again, now we're narrowing that focus a little bit more specific. And now we're just looking at variants in the ALK gene. And so you can see here, they come up in this large table. There's still even a lot of them. So we've gone from millions down to 60,000, which is still quite a large number. But each of these variants is given with the genomic coordinates. If it's associated with a DB-SNP allele, it has that as well as the specific genetic consequences. So these are premature stop codons that have arisen in these particular variants. And then over on the, as we continue farther, it gives us additional information about the frequency of the variant. And so these are, only one participant in our Kids First data set has this first one up here, but you can see this one is very, very common. It's much more frequent. So you could, in fact, then let's say maybe we want to learn more about this one. So we'll click here. It gives some additional information. So we have genetic impact that's scored with Kat and Dan, as well as VEP annotation. So this is predicted to be a high likelihood variant of having a biological effect. It gives us information about the different studies that the variant was detected in, as well as any clinical data associations that are known about this particular gene. In fact, it's already known, as I alluded to, ALK is a widely known gene that's associated with neuroblastoma. So that is linked here and indicated there as well. If I go back to the variant search, that particular variant is also linked out to DB-SNP. So if we can click this, we can learn more about the record for this variant, to learn more about this this particular gene, its frequency and populations, et cetera, et cetera. So from this point, as I showed, I can go back to this main tab with our frequencies here. This gives us information about particular participants that have this variant. And then we can say, oh, they're interesting that participants have this ALK mutation also in the orofacial cleft European ancestry study. You can then select this cohort, and then it will pull up that set of participants for you to be able to analyze further. So at this point, you can then select those files as well. All right, so that's 3 different ways to be able to search data in the Kids First portal. So now I'm going to move into our second file. So now that we've found our participants, how do we identify any files or genomic data that is relevant to them? So I'm going to go back to our first example here. So our Kids First neuroblastoma study, and it has 18,000 files, which is a lot. So let's take a look at them and maybe narrow them down a little bit. So when you click that link, it takes us to our 5th tab up top. So now we're in the file repository. So if we searched based on clinical data in Explorer data, and we searched based on genetic data in the variants in the file repository, now we're searching based on files. And so we're looking up for specific files, and we're trying to narrow those files based on specific file filters. And so those participants that we moved over from the previous tab are showing up here, as well as the number of files and number of families. We can narrow that down if we choose. For example, if we're just interested in the whole genome sequencing data, we can select whole genome sequence, and it will add another tag here, another filter, which will then narrow down our data set quite a bit. And what we could imagine if we're interested in running our own analysis, we could start with the alignment files. So we would start with those in cram format. And you can see now we're getting smaller. So now we have 1,300 alignments and 1,300 participants. So we're getting closer and closer here. At this point, we could continue to narrow it further if we wanted to select different clinical filters. We could apply those as well, but I'll end the example here. So at this point, how do we actually access these files? So right now it's just a table. How do we begin to use these as part of our analysis? So step 2B, we're going to apply for access on dbGaP. And so each of these Kids First studies is enrolled under an NIH process called dbGaP, which is what allows investigators to be able to apply for access, receive approval, and then access the data on the Kids First portal. So I already have access, so I have a little download button here at the end. But if you don't have access, you'd see a little lock symbol. So how do you unlock the lock? You unlock it by applying on dbGaP. So this is where these PHS IDs come in. And so you can actually click this PHS ID. And it sends you to the dbGaP page for that particular study. So here we're looking at the human neuroblastoma study within Kids First. Provides some additional information about the study. So this is provided by the original principal investigator who consented the patients. We have some additional information about what type of consent the patients enrolled when they joined the study and what types of research the data could be used for, as well as any publications from the study, as well as a list of other investigators who have requested access. So from here, you can then request access and then submit an application through NIH to be able to receive access to the data. Access to the data is free, so there is no fee for accessing the data. There's no fee for applying. There's no fee for downloading the data. We're a publicly funded, publicly supported program, and so everything is open access and available. We are making it available to everyone in the research community to access. Data access is free. Data access is also available to everyone. So yes, this is based out of the United States, but investigators anywhere around the world can apply for access to the data. So this is not a limited based on where you are an investigator. Anyone can apply and submit applications. So we have documentation on our website. I won't go through actually logging into dbGaP and submitting the application, but this would be the site that you would be able to use. Additionally, on dbGaP, you can combine multiple data sets into a single application. And so investigators can, if you see multiple studies here that you're interested in combining for your analysis, you can list multiple PHS IDs in your application, and they'll be considered together for combined analysis. So you submit that to NIH dbGaP, and they typically turn things around within 48 hours. And then you'll get an email that indicates, congratulations, you've been approved, hopefully. Once that's done, then you'll want to go into the Kids First portal and connect your account. Basically what that does in our settings tab, it will allow the Kids First portal to be able to connect and see that you've been approved, kind of like checking your passport at the airport, so to speak. So it sees that you're authenticated to have these files, and then the little locks will unlock. So like I said, I've already done all that because I am an employee here, and so I already have this action. So I can, if I'm interested in working with this file, I could even click the download button, and then you can see I'm beginning to download this file. So this is a very big file. This is 20 gigabytes, and there are 131,300 of them. So I think I would quickly run out of hard drive space if I did this for every single file. So I'm going to cancel this download. And we're going to work in the cloud to be able to do our analysis. And so I'm going to select, rather than selecting all of them, I'll select maybe just the first 5, 3, 4, 5. And I'm going to analyze them in Kivatica. So again, Kivatica is our cloud-based analysis platform. And so rather than work on them on my computer or on a local local high performance cluster, I'm going to work on them in Kavatica. So I'm going to copy these over to a Kavatica project. I already have a demo project set up here, St. Baldrick's demo. That's where I'm going to copy them over to. It says success. The files were copied to your Kavatica project, St. Baldrick's demo. Hooray. So now they're available for analysis and investigation. All right. So then we've achieved step C there. We've connected and we've pushed to Kavatica. So then in my last couple of minutes here, I'll show a little bit about analyzing data in the cloud on Kavatica and what that looks like. So there's actually a link here when it popped up that I could click directly to enter there. But rather, I'm going to navigate to Kavatica by going to kavatica.sbgenomics.com. And again, I'm going to log in with my ERA Commons, but anyone can create a username and password with any email address. So you don't need an NIH ERA Commons to log in. So Kavatica is organized around the concept of projects. And so when we log in, we can see here, this is kind of my homepage where I have a list of all of my projects that I'm a part of, and then a list of the analysis that's been going on within those projects as well. And so I'm involved in a lot of different things at the center, as you can see, but we have a set, our demo space here, St. Baldrick's demo, there's that project that I created earlier today. So I'm going to go in here to St. Baldrick's demo, and this is an overview of what the project looks like. And so on the left-hand side, there's a workspace. You can kind of like a whiteboard. You can add a specific description. You can write notes to yourself. You can write a to-do list, or you can highlight specific functions. Kind of a nice little area for you and your collaborators to work and write. On the right-hand side, this is a list of the members. So right now I'm all by myself. I'm all alone. But if we wanted to collaborate with investigators, I could invite new people. So for example, if I wanted to work with Jan Kuhn, who's on he's the lead of our bioinformatics unit, I could type his name and invite him just by clicking and then invite him to the project. And then he would pop up and he would have access to the same files and resources that I'm saying. It also lists our analyses. And so you can see here, I ran an index last month, as well as a genotyping workflow, which I'll show a little bit more in a minute. So moving across other types of things that are available in Kavatica. So I moved those files over. You'll remember I selected these 5 files and then I analyzed them in Kavatica. So where did they go? So they landed in the Files tab. And if we click over here, aha, you'll see there's my 1, 2, 3, 4, 5 alignment files that I selected out of the Kids First portal and then pushed to Kavatica for analysis. So at this point, each of these are, they're cram files, so they're very, very large and they're compressed. And so right now, there's nothing a whole lot to do in terms of preview just by looking at them. So maybe I need to do some analysis on these crams in order to be able to extract output. To do that, you want to use an app. And so apps are the next column here. So apps come in a couple different manners. So you can add an app from our list of public apps. So this is a bit like the App Store on your phone. You can search for existing applications and then try to add them to your project. And so, for example, we can type in GATK, and you can see a number of the GATK processing tools are already available here that we can then apply in order to be able to run our processing. You can also create your own app. And so if you're interested in bringing your own tool, you can. And so if you have your own locally developed workflow, especially one that is Dockerized or written in the common workflow language, it's very easy to import it into Kavatica, where you can run it in the cloud as well. And more details about that are available in our help documentation, in the Kavatica documentation as well. Once you have a particular tool, so for example, maybe this indexing tool, It provides information about different inputs, different outputs, and allows you to be able to set up a run. So once you try to run an app, that generates a task. So a task is one instance of an app. So this is an indexing for BCF tools. At this point, then it provides different inputs that are defined by the app. So this one, I want to input variant files. And so I would select them here. I can then choose different settings. And then when I run the tool, it would save the output. So I'll show I can select the files. And even though, here it's limiting me. And so it's looking that I'm expecting that I would only be applying a VCF. And so my params aren't showing up here because this is the VCF tools indexer, but you can select files. We can add them to the project, and then they appear here as our input. We can, if we want to apply additional, values and additional kind of curation of the tool, we can do that in the middle, and then we would eventually put it in the run. And so I'll show you what. So this is what it looks like before you submit it, but I'll show you one after. And so our BCF tools output. So this is a run that I ran in November last month, where I was generating an index of a BCF file. And so what's great about Kavatica is that it actually tracks everything as well. And so even though I ran this a month ago, I have all of my information here. This is my input. This links to my output. It gives me information about what settings did I choose. And if I want to run the same exact thing again, I can click the blue button, edit and rerun. So it allows for reproducibility and repurposability of tools and data sets to be able to process. And then finally, so once I have these files, I really want to take a look at them. And so the Data Studio is a great place to do that. So the Data Studio is a analysis suite that allows users to be able to run either a Jupyter Lab environment with Python-based notebooks or an RStudio environment to be able to look at the data, really dive in. And so it has the same format as Jupyter Lab and RStudio that you might have on your desktop, but now you have access to all of these files and you can bring them into your analysis very seamlessly and easily. So it allows for additional, generating that first analysis, generating those graphs, and then that you can use those, directly into your publications without ever downloading any of the data. So it's all available in the cloud through these interactive analyses. All right. And so I only have a couple minutes left here, and so I will wrap up very quickly by sharing some additional places to follow up and learn more. Here we go. All right, so if you have more questions about Kids First, then I would encourage you to check out our Kids First office hours. These are monthly help sessions that we host. And so I'm there and available, as well as members of our data analysis and our bioinformatics teams. And we're available to help answer questions, everything from, what is Kids First? How do I get started? All the way to, I've done an analysis, help me, what do you think of these results, type of stuff. These are hosted on the second Tuesday of every month at 3 o'clock Eastern time. And so in fact, that means our next one will be next Tuesday, 1 week from today. If you're interested in learning more, please check out our social media page or visit our tiny URL for more information and links on how to join. I also want to emphasize that the NIH is currently sponsoring a cloud credits program to support projects on Kavatica. And so what this means is that NIH is offering investigators credits to be able to run their analysis and get started. So this is a really great time to begin working with Kids First data and using it as, as to support your efforts, because NIH will help you, provide these cloud credits to be able to run the analysis. So these allocated credits can be used for research projects using Kids First data sets, as I've shown. It can be used for combining your data with Kids First data sets as well, or for developing software tools that you're interested in analyzing Kids First data sets on. Each investigator will receive a $1,000 allocation, which is available to help get started on the platform, and then larger allocations could be available with demonstrated need after testing workflows. If you're interested in learning more, I encourage you to check out our repo with some documentation, github/kids-first/kf-cloud-credits. And also, or you can email the Kids First program, kidsfirst@od.nih.gov for more information. If you do a internet search for Kids First Cloud Credits, you'll find information that you're looking for. All right, and so that's my final slide. So once again, I want to thank the organizers here at St. Baldrick's for inviting me to attend today. Again, I want to acknowledge all the wonderful people who've helped build the Kids First data resource and make it such a wonderful place and a wonderful tool and platform that we're making available for free to investigators around the world. So with that, I'll wrap up. And again, encourage you to check us out and visit us at kidsfirstdrc.org to learn more. You can follow our social media at Kids First DRC on all major platforms to learn about events and training opportunities. And I encourage you to reach out to support@kidsfirstdrc.org if you have any specific questions and are looking for help getting started. And of course, thank you very much for all of your attention today. Look forward to helping to support your research in the future.